So, today we are going to actually be kind of reviewing the last unit as well as combining it with this one. So we're going to be talking about our organic compounds and how they kind of play into all of the organelles. We're also going to talk about a little bit of specialization. We're going to answer this question today about how organelles help the cell to function and how specialization plays into that. So the first thing, with your partners, write down the 4 organic molecules that we discussed in the last unit. My computer, I need to mute it. There we go. So yeah, we had 4 different ones. Can anybody tell me one of them? Lipids. Carbs. Proteins and nucleic acids. Yes, good. So we're going to be talking about those 4 different molecules. We're going to kind of hit on them a little bit again, as well as talk about, like I said, how they kind of play into all of our different molecules. So the first one we're going to talk about, actually that was the question, there we go, is carbohydrates. So carbohydrates, anybody remember what the use of carbohydrate was? Short-term energy. Good. So these are going to be utilized in our mitochondria and our chloroplast. So again, with your partner, look at these 2 reactions on the bottom. We have photosynthesis and we have cellular respiration. Look at the 2 equations. Tell me how they are similar or different right on your boards. Okay, so anybody want to read off what they thought? Liam.
Photosynthesis uses what cell respiration makes, and cell respiration uses what photosynthesis makes.
Okay.
So it's like a cycle.
Okay, so it's a cycle. So we have carbon dioxide and water on one side to make glucose and oxygen, and on the other one we have glucose and oxygen makes carbon dioxide and water and energy. So any other ideas?
The same elements are combined for a different outcome.
Okay, so it does make a different outcome. So again, we have glucose and we have oxygen on this side, and on this side we have carbon dioxide, water, and energy. What about the energy? Where is the energy in photosynthesis? The sunlight.
Yeah.
Yes, I heard someone say.
Right.
So the sunlight is going to provide energy. That's why plants absorb the sun's energy through photosynthesis in order to make these. On this side, we have this weird ATP molecule. We'll learn more about that next unit. But this is going to be the energy of the cell, this ATP. You can see that on this one, the light or the energy goes into the reaction to make the products. For cellular respiration, it is spit back out as a product. So, yeah, these reactions are both going to use carbohydrates. So they use glucose. So if you don't have that one in your memory, this is one that you will want to remember. Glucose is C6H12O6. If anybody remember the ratio of carbon to hydrogen to oxygen? 1 to 2 to 1. What do you see here? 1 to 2 to 1. So glucose is going to be the sugar that we produce from photosynthesis and the sugar that goes in for cellular respiration. But you can see it's literally the same reaction backwards. One is putting energy in to make sugar, that being photosynthesis, which is going to happen in your chloroplast. The cellular respiration is going to break down that sugar and form our usable cellular energy in the form of ATP. And that will be done in the mitochondria.
He's not sitting in his seat.
He's working. Same thing with Matt jumped back.
Yeah.
So yeah, the cell walls are made of cellulose. So the cellulose, like I said, is going to be very structurally supportive since polysaccharide is really long, longer than this up on top of the board. And like I said, it's going to keep the carbohydrates very sturdy, help it maintain its shape. So that's why they also look very boxy, is because of this these long chains. So here's another question for you to talk out with your partner. Why do we use carbohydrates? Why do we use a polymer made out of glucose? So write down on your board why you think that it would be made out of glucose. Okay, who wants to read off their answer? Abigail?
They make glucose from photosynthesis and they produce a lot of it, so then they use it to keep the cells and cell walls in shape and place.
Good. So yeah, in photosynthesis, the last slide, we saw that glucose and oxygen were a byproduct of photosynthesis. So yes, they do need to use it for energy, but they're making so much glucose that they can use what they don't need for energy to help support their cells. So they do this by combining all of those molecules together to form cellulose. And that cellulose can help repair damaged cells, it can help make them more sturdy, and it can just help to overall benefit them.
So if for whatever reason they start to make less glucose, does that mean the cell deteriorates? Might deteriorate.
Yeah, so they're not going to be able to do maintenance as easily to fix all of those. Yeah. So any questions about carbohydrates? Okay. Now we are going to move on to lipids. So, when we talked about lipids last time, we said that there were 3 different polymers. Well, polymers. We said there were fatty acids, or saturated fats, not unsaturated fats, and the third one that wasn't really touched on a lot was the phospholipid. So the phospholipid is now going to be very important, because phospholipids are different than the ones we had talked about. These only have 2 tails. The ones we had talked about before had 3 tails. And instead, where one of the tails would have been, there is this phosphate group up on top. There we go. So up on top we have this phosphate group. And this phosphate group is going to act really weird. It has a lot of different structure, or a lot of different oxygens on it. Some of them don't have some of them have a negative charge. So what happens is this guy is actually polar. So you have a polar head, which is this red bit up here. So all of the glycerol and all of the phosphate group are this hydrophilic polar polar head. There we go. And down here, the tails, one of them is an unsaturated tail and one is a saturated tail. They don't like water. So this helps to create what's called a lipid bilayer. So bi means 2. So the phospholipid is going to point with its head towards the water and its tails out of the water. So they kind of line up like this. So the tails are in the middle and the heads are on the top. And this is going to help create that cell membrane that we have around all of our cells. This also helps to allow the membrane to be selectively permeable. So we talked about permeability yesterday. Anybody want to say what permeable means? Anybody remember what it means?
Able to be is it not dissolved, but kind of dissolved or mixed? I don't know.
Kind of. You want to help them out?
Able to move through something?
Good. Yeah, it's able to move through it. So selectively permeable means that only certain things can move through. So for example, water, because it is a polar molecule, it can't get through this big wall of tails, so it's stuck on the outside unless we use things like pumps. On the other hand, we have ions that also have to go through those pumps, but things like, oxygen are not charged. So oxygen doesn't have a charge, it's just two oxygens stuck together. They're also small enough that they can fit through the cracks and just go into the cell. There's no need for a pump or anything. They can just sneak right through into the cell. So that is what we mean by selectively permeable. Some things are kept outside, some things are kept inside, and others are just allowed to move freely between them. And there's one other thing, even though it's not written up here. When we talked about our saturated and unsaturated fats, we said that saturated fats are going to have those straight tails, and they are going to be able to be really solid. They can just kind of stack on top of one another. We said that unsaturated fats were, because they have that kink in the tail, that kind of double bond that makes that little kink, they are much more liquid. So, if we are using a saturated fatty acid and an unsaturated, or an saturated tail and an unsaturated tail, talk with your partner. What do you think the plasma membrane is going to be more solid or more liquid? So again, think about the saturated fats versus unsaturated fats. Do you think the plasma or the cell membrane is going to be more liquid or more solid? All right, let's come back together. So anybody, first of all, hold up your boards. So we have a lot of liquids. We have some kind of half and half. So someone who said purely liquid, do you want to tell me why? Why do you think it would be just liquid? I'm going to pick on the side that chose both too, so it's not like I'm just picking on one side. Does anybody want to share why they think it's going to be liquid?
Because water is let through.
So water is going to only be let through pumps. It can't slide through these. It's kind of like if I hold up this jar, the layer between the blue and the, actually let me hold it over here. The layer between the blue and the oil would be like this plasma membrane. It's too thick for it to get through.
Okay. Solid.
Okay, well, liquid was a good idea. Think about how the tails are arranged. Do you think these can stack on top of one another well?
Yes.
No, because they have one normal tail and one kinked tail. So they can't really stack on top of one another. What if we had two unsaturated tails?
Then they would be able to.
You'd think they would, but they would have to have something else keeping them in the same orientation. So they'd all have to be facing the same direction. So otherwise you'd have one that's facing like this, one that's facing like this, and they wouldn't be able to stick together.
Okay.
So these are slightly, well, actually, before I get to that, the side who chose both, what's your justification for it? Why do you think it?
Well,
Partially straight.
well, yeah, like you just said, like since they can't like stick, then it wouldn't be solid. But since they're not totally opposite of each other, they're not liquid. So
Okay.
so like when you're in between, it gives something that is like the top part of like before it cuts off to that weird angle for the unsaturated one, they kind of like connect.
Yeah, so in reality, the cell membrane is somewhat liquid, as we saw in that one animation, but it's not liquid enough that it's just going to turn into oil. So it is kind of in between. It's it's fluid enough that it's going to be able to like move around. It looks kind of like water rippling, but they are strong, or they are straight enough that they are going to actually still stick together. Okay. So any questions on lipids? Okay. So we are going to continue on with the plasma membrane. And now we're going to talk about those proteins. So I said about pumps, and these are going to be different proteins that are stuck into the cell membrane. So yesterday when you played your cell identification game, there was that little channel thing that we hadn't talked about. And those are called protein channels. So you have a picture on the right side of your thing that shows different protein pumps and different protein channels. But proteins are going to help regulate what goes in and what comes out of the cell. They're going to be like elevators. Elevators and like security guards. They're going to say, okay, this molecule can come in, this molecule can go out. So for example, water is regulated through a protein pump. It's going to go through a specific channel that allows it to get into and out of the cell. There's also going to be cell receptors that stick out of the membrane, and that's going to signal things like, oh, a certain hormone needs to be produced, and then that'll trigger in the cell, or different sensory things. But proteins are going to be important because they're embedded into the cell membrane. And like I said, they're going to help get things in and out. Any more time? Okay. You all good, Peter? You got everything?
Yep.
Okay. So, the next part we're going to talk about, is jumping away from the cell membrane, are the ribosomes. So the ribosomes, while they aren't exactly organelles, they are going to be extremely important because they build proteins in the cell. So these include a lot of different molecules. For example, structural proteins are going to be made. Different hormones will be made. Cytoplasmic proteins, so all of those pumps and things, will be made using ribosomes. Also, enzymes. So if you remember, enzymes are a type of protein. They are not made from nucleic acids. Instead, they are made from proteins. So that was a question on your test. But enzymes are going to be made using ribosomes. So and actually here, I'll let you write and then I'll ask another question.
Do you have a test tomorrow?
No. I think I'm going to put the quiz on Monday just so we have more time to review. Also, I keep getting yelled at for having tests on Fridays, so it's just timing. Just timing.
I'd rather have a test on Friday than Monday.
It's more fun to review, though.
My dad would be so much fun. I love when she gives us reading every single day.
All right. So, your next question. If, for some reason, ribosomes stopped working, how might they affect the cell? if ribosomes, the protein builders, stopped working, what might happen to the cell? No. No. So, who wants to give their answer as to why they wouldn't work? Noah.
So the cell would stop letting things in because the proteins is what lets things into the cell. So, like if something is like broken, it can't be fixed.
Okay, so it would stop letting things into the cell. So as our protein pumps and things like that would stop working, the ribosomes aren't there to make more of them. So it wouldn't be able to help let things into the cell. Any other ideas? Liam?
I don't know if this is important, but we covered it yesterday. Without these proteins, there would be a lack of enzymes, and then without the enzymes, there wouldn't be the lysomes to clean up the cell.
Okay, so no enzymes. So not only would the lysosomes not work,
Yeah.
every reaction that happens in the cell, if it needs an enzyme, is it going to work? No, because there's no enzyme to speed up the reaction. They can't reduce the energy. So if there's no ribosomes, then there's no enzymes, which means there's no reactions. Well, not as many reactions.
That's not good.
Yes, which is not good. So yeah, any questions about ribosomes and protein building and all of that?
Nope.
Okay. The last one we need to talk about are the nucleic acids. So nucleic acids are important because they make up what?
DNA.
DNA, as well as
RNA.
RNA. Good. So DNA is going to be stored in the nucleus, as we said a couple of days ago, and it's going to be stored as chromatin. So that is the term we use for the loose DNA. So I grab my wonderful little model. This would be chromatin. So it's stretched out, it's nice in its double helix ladder shape. If we want to condense it, especially during cell division, we will combine them. Actually, it wraps around a protein, but in this case, I'm just going to collapse it. And it's going to create what's called a chromosome. So you've probably heard of chromosomes before. But chromosomes are going to be the kind of collapsed, condensed versions of our DNA. So chromatin is really long and stretched out. Chromosomes are nice and condensed, and they form this little X shape. So nucleic acids are going to play a role in both of these. So they are both made of nucleic acids. So all of our genes are going to be determined by the nucleic acids. All of our different personality traits, all of our characteristics are all going to be determined by what base pairs, that A, T, C, and G that we talked about, are present. It's also important for RNA because RNA needs these different nucleic acids to code for different proteins. So we'll see whenever we get to translation and transcription, this is going to be really important because different letters mean different things. If we have, for example, a U, or an A, a T, and a G in a row, that means that a protein will start to form. If you have that specific letters. If you have A, C, G, doesn't work like that. So we'll learn whenever we get to translation and transcription that nucleic acids are going to be very important for what proteins we're making. Set down my wonderful little chromosome. So are there any questions about nucleic acids? Any questions about any of the molecules, both from last unit and from this unit? Okay. Does this make sense? Now you're starting to see where the actual so we're not just learning about the molecules, we're learning kind of what they're used for. So hopefully this makes sense comparing like what we learned last unit and kind of applying it to how it affects the cell. Does anybody need more time writing? Okay. So, the last question I'm going to ask you. How do cells work together? So discuss it with your partners. Write down a list of ideas that you think might help cells to work together. Let me just pull up. So, like I said, the LEQ for this lesson was how do cells help, or how do organelles help a cell to function? So what do we think, how do they work together? Anybody have an idea? So they create different things, okay. Any other ideas?
So,
Peter and then Abigail can go.
okay. So certain organelles create stuff which other organelles use, and then it's basically just a chain. What one organelle does helps another organelle do what that does, and it's basically just like a loop between all these different organelles, and then it all just loops back to that organelle.
Okay. So collaboration.
Yeah.
So all the cells work together to achieve something.
Yeah.
Any other ideas? Oh, Abigail first, and then, sorry. Abigail, and then Emily. So they do help to maintain homeostasis. So there's a couple different things that they can do that help to kind of regulate themselves, regulate our bodies.
They're given specialization, so like a stem cell could be used for then like skin cells or heart cells, like cardiothoracic
Good.
cells, so then they can be used and cohesive in the body.
Mm-hmm. Yeah, so cells will give each other jobs. So kind of along the lines of that collaboration, these cells will kind of designate jobs to one another. That's not what I wanted. So, in terms of specialization, yeah, certain cells will give each other jobs so that they can work. So, during your microscopy lab, when you looked at the onion cells and the aquatic plant cells, what did you see different between the two of them? This was one of the analysis questions. So
Aquatic plant cells had like a chloroplast.
they had chloroplasts. Why?
Because they the sun.
Because they maintain homeostasis.
So the homeostasis kind of. This is more along the lines of like giving each other jobs. So what was it?
The sunlight, they can like see the sunlight, they can see the sun. So
Which ones?
the plant cells of the aquatic plant.
The leaf cells. So in this case, we are specializing our different parts of our organism. So the leaf cells of a plant have a lot of chlorophyll, a lot of chloroplasts that help them to do photosynthesis. If we look at root cells like onions, they're underground. How much sunlight are they going to get? None. So do they need the did they need the chloroplast to do photosynthesis? No. So by specializing, by giving things different jobs, it helps the organism to run more efficiently and to do things like maintain homeostasis. So homeostasis, like I said, is going to be that maintaining the internal environment. So doing things like producing hormones or releasing water or different things like that are going to help these cells, and if depending on whether they're unicellular or multicellular, are going to help regulate that organism. They're going to be able to survive a whole lot better and adapt to change a whole lot better. So any questions about any of this? I'll give you a couple more seconds to write all this down. But any questions about what we've covered today? Okay. So, for the rest of the period, you can continue working on that activity board. If you have something that still needs to get done, please make sure that it gets done. Like I said, it's due by tomorrow. Tomorrow we will probably review some more. I have a couple more stations I'm going to do. So, yeah, with that, you guys can get to work.